Hi everyone. My name is Ido. I'm a student at Professor Adishtan and Professor Talpuk Labs. Just a moment. Okay, I will present a research about harnessing machine translation method from the NLP domain for the sequence alignment problem. This is one of the fundamental problems in bioinformatics field. This work was done with the collaboration of Dr. Jonathan Beniko from the Technion. I'm quite excited. This is my first time as a speaker. So let's start with the problem. The sequence alignment problem is known to be an NP-hard problem, and many biological tools uses the alignment as an input. Without aligning sequences, one cannot do anything in the bioinformatics world. So in biology, a DNA sequence is a sequence over the alphabet of 4 letters or 4 nucleotides, which we symbolize by A, C, G, or T. Those sequences contain the genetic code, which later will create the protein inside the cells. Now, consider sequences from a common ancestor, and the sequence alignment problem is once we pass those sequences through alignment algorithm, we expect to get something like this. In our world, something like this, where we identify regions of similarities between those sequences. In our world, we consider 2 types of events. The first one is substitution, like we see here, where the E transpose to G or the G transpose to A. We don't know which one of the events happened, or something like this, where we have insertion or deletion. Maybe the G was inserted to the second sequence, or maybe the G was deleted from the first sequence. As we don't know if it was insertion or deletion, we call this event indel. For example, if we want to think about when using the alignment problem, consider that we have a viral genome from the coronavirus, and we want to identify if the genome refers to what variant. Is it alpha, beta, gamma, delta, or omicron? The first stage will be to align this genome to the profiling of all the variants and find the profile with the least distance. So in the previous slide, we saw an example of 2 input sequences, but of course we can generalize the problem to many input sequences, as you can see here. Here we see 5 input sequences. We have a substitution example here, and we have a indel as well. As we increase the number of sequences, the problem becomes much more complex. So now let's talk about machine translation method. The transformer is trained to translate sentences from an input language to an output one. So here we have a transformer that was trained to translate sentences from French to English. And we wanted to harness this method for the sequence alignment problem. So what we did, we considered the input sequences as an input language, and we considered the alignment, the result, as an output language, and then we trained the transformer to translate the unaligned sequence to the aligned sequences. When we did it, we need to consider 2 things. The first one is how to encode the input sequences to one sentence, because the transformer translates sentence to sentence, and once we translated the sentence, we need to decode the result sentence to an alignment. So once you have the encoding and decoding, you can train transformer to align the sequences. So now let's check our result compared to the aligning methods today. So this graph represents the error compared to the number of input sequences. We compared our result to MAFFT and Prank. Here you can see the error of MAFFT. MAFFT was published 20 years ago, and it has many sites. This is consider only the site of the first version. There are, I think, 7 or 8 versions of MAFFT. And as you can see, as we increase the number of input sequence, the error increases as well. About 15 years ago, a new software was published called Prank. It was published in Science, and it had a better result compared to MAFFT. In most cases, Prank reduces the error. And now you can see our results. We have, in most cases, superior result to MAFFT and Prank. And we are talking about reducing the error compared to MAFFT by 25 to 35%, and compared to Prank about 10%. So we have superior results. But let's talk about other cool things that we can do when using machine translation method, when using deep learning. So today alignment software are using many input configuration and thus have an assumption about the alignment dynamics. In practice, everyone uses the same default parameter. So when running those programs with the default parameter, if this is the alignment dynamic space, when using the default parameter, you have a sub-region of the alignment dynamics. We know for fact that different species have different alignment dynamics. So each species is in a different sub-region of the space. When we are using machine translation method, we can train different transformers in each of the sub-regions of the alignment dynamics, and so the transformer learn to learn the sequence without any previous assumption, but learns it straight from the annotated data. We don't need any input configuration because we can just train in those specific sub-regions. Another cool byproduct of this deep learning approach is using the embedding vector of the unaligned sequences. So the transformer is composed of 2 parts, the encoder and decoder, and there is high dimensional vector that passes from the encoder to the decoder. In our case, this vector contains about 200,000 features, and this vector is a good representation of the input sequences. So we took this vector and we trained different machine learning tools to predict other things. So you can see the result of a simple linear regression of this vector to predict the length of the common ancestor. We had a pretty good R-squared value. Another cool thing that we done is we measure the effect of the transfer learning. To achieve our results, we started with a simple dataset 1, and we slowly increased the complexity of the alignments. So dataset 2 is a bit more complex than dataset 1, and so on. So the transformer that trained on dataset 1 was the initial transformer when we trained on dataset 2. After training on multiple datasets, we return to dataset 1 and we measure the results again after training on multiple datasets. So the left bar here represents our initial error, and the right bar represents the error after using the transfer learning. So you can see we reduced the error by about 25%. So we can say that in our case, transfer learning increased the performance. So here you can see attention map from NLP classical task. Our attention map is quite different. You see that here most of the attention is around the main diagonal, but here we have many diagonal as well as the attention map is much larger. So what I'm trying to say is that the NLP method was good, but we need to develop new method in order to support biological sequences. And so this research took a different approach to solve a long time problem in biology field. We use cutting edge technology from the deep learning field and especially from the NLP domain. And we believe that most of the tasks in bioinformatics could be solved with NLP method, as many things in biology are based on sequences. I would like to thank everyone who took part in this project, especially Dr. Jonathan Belikov from the Technion and both of my mentors. So thank you, and if you have any question, you can ask now or you can contact me in the mail.
Thank you. We have time for a few quick questions. First of all, have you compared transformers to simple neural network architectures?
So we did compare different architectures of transformer. We tried running on BERT and different architectures, but it's not so easy to compare the transformer to LSTM. But we will do other architecture as a few weeks ago. It was published a new architecture called S4 to support longer sequences. So we might try it as well.
Okay, another question. Did you consider adding another feature to the network, such as the species type, instead of training dedicated transformer for each species?
So if we have unaligned sequences, we have tools today to extract alignment dynamics from the unaligned sequences. So it's not important the species, but it's more important the parameter itself, and we can train on different ranges of the parameters.
Okay. One last technical question. The example that you gave with the transfer learning, which data were you using? This is again multiple alignments or something else?
Yeah, so this is again multiple alignments. We start training on dataset 1, and then we
and the different datasets are just unrelated alignments?
Yeah, so think about dataset 1 as pairwise alignment, and then dataset 2 maybe three input sequences, and dataset 5 maybe 10 sequence alignments.